Hello students, welcome to EPG Pathshala. I am Mohan Chandan Joshi, Assistant Professor from Jamia Millia Islamia. Today we are going to talk on module Gateway Cloning under the paper Genetic Engineering and Recombinant DNA Technology Part 1. After completing this module, you should be able to understand Introduction to a general cloning method. What are the cloning methods which are generally used in the lab to clone a gene of interest? Then in the second part you will be able to understand what is gateway cloning. And third, you will understand what is BP and LR reaction which are involved in gateway cloning methods. So what are general cloning methodology or cloning methods that are available for researchers today? The very first one is restriction enzyme based cloning. This is very commonly used methodology in the lab, in which restriction enzyme cut site in combination with restriction enzymes are used to clone your gene of interest into a vector. Second is TA cloning. TA cloning is based upon the PCR product that we get after PCR reaction in presence of a tagged DNA polymerase, which results in an A overhang and can be easily cloned into a vector which has a T overhang. The third is a ligation-independent cloning method, in which we don't require a restriction enzyme or DNA ligase activity, but a multiplex PCR which allows, and in combination with T4 DNA polymerase, we can ligate our gene of interest into a vector without any restriction digestion or restriction enzyme usage. Fourth is a multicistronic cloning method. This method allows us to clone not only one gene but 2 or 3 genes in tandem, which can be driven by, and the expression of these genes can be driven by either a single promoter or can be driven by independent promoter. Last and least is recombination-based cloning system. Gateway cloning is part of this recombination-based cloning method. There are two methods in it, two methods that are available commercially right now. One is infusion cloning and second is gateway cloning. So we'll talk about gateway cloning in detail in next slides. Restriction enzyme based cloning approaches. This is most commonly used cloning method in a molecular biology lab. The essential requirement for this cloning is that your gene of interest should contain a restriction enzyme cut site, which should be same in the vector which you want to clone in. In case your gene of interest does not contain that restriction enzyme, you can introduce this restriction enzyme site into that DNA by amplifying it using the PCR primers with an overhang of those restriction sites. Restriction enzymes are enzymes which cut DNA sequences, and they are either 4-base cutter or 6-base cutter. If they cut both the strands at the very same site, they result in a blunt end. They are called blunt end cutters. And if they recognize 2 different sites on the double strand DNA, then they result in a 3 to 5 prime overhang, or also known as sticky end, and these enzymes are called sticky end cutters. Both blunt end cutter as well as sticky end cutters can be used for cloning purposes. However, most convenient to use are sticky end enzyme cutters because they leave 3 prime to 5 prime overhang, which can be religated by DNA ligase enzyme. Restriction enzymes cut site in your gene of interest as well as in your vector should be unique. It means that your desired restriction enzyme cut site should not be present in the gene of interest, otherwise while digesting or preparing your vector as well as insert for cloning could result in multiple bends. TA cloning. TA cloning method exploits the ability of tagged DNA polymerase to generate an A overhang at the 3' end of the DNA after PCR reaction, because that lacks the exonuclease activity or proofreading activity, which would allow it to generate the blunt end. On the other hand, if we use a high fidelity DNA polymerase, which is PFU, it would result in a blunt end and would not be used for this particular cloning approaches or this cloning method. So as you see in this schematics, a PCR product is generated, and that PCR product could be our gene of interest, which we can amplify by using specific primers, and the eventual PCR product will have A overhang. In this we have a vector which contains a T overhang, and as you know, A and T are complementary to each other. If we ligate these two products, our PCR product as well as the vector which has a T overhang, we would clone our gene of interest at the target site. Ligation-independent cloning method. Compared to the previous two methods, this method does not require any ligation approach or any ligation method. As you remember, in a previous two slides we discussed about that our gene of interest is ligated with vector. In this method we exploit the 3 to 5' exonuclease activity of T4 DNA polymerase. The DNA is our gene of interest is amplified by using PCR, and now this particular PCR product is incubated with T4 DNA polymerase, which generates cohesive ends because it has the 3' to 5' activity. And if this activity is allowed to continue, it can chew up the entire DNA. Therefore, we add DNTPs, which will stop the exonuclease activity of this enzyme. Now we will have the vector which will have the similar cohesive end which were generated by using similar methods. So by annealing, by incubating these two products together in presence of a T4 DNA ligase, we will have our gene of interest cloned at the site at the vector. Multisonic cloning. This strategy allows us to express 2 or 3 proteins from a same vector, and this is often required in an experiment where you want to keep the copy number for our gene same. However, in this case we can have two strategies. One, that we express all these genes, whether it is 1, 2 or 3, under the same promoter. On the other side, we can also have independent promoter for each of these genes. We need to make sure that if we have an independent promoter for each gene, that each promoter has a Shiner-Delgado sequences as well as the start codon. Otherwise, if we are driving it from a single promoter, we'll have just one SD sequence and one start codon for that. Multisonic cloning approach. As in the previous approaches which we have discussed, we had a single gene of interest which was cloned into a vector and subcloned into some certain other vector for our desired molecular biology application. However, in case we want expression of 2 or 3 genes simultaneously, which is under the control of same promoter or as an independent or under an independent promoter, we can do that by using this approach. In this, we can have 2 or 3 or 4 genes cloned in tandem, and their expression is driven by either single promoter or individual promoter. This particular cloning system is very useful or very essential in a molecular biology application where we want that 3 different proteins are expressed at 3 different level or at a variable expression level. So we can manipulate that by having 3 kind of promoter for each of these genes. Just a simple schematics of how 2 genes could be simultaneously cloned into a same vector. Here is the schematics of that. In this, you see 2 different plasmid representing as block A or gene of interest A on one side and gene of B as a block B on the other side, and there are 3 restriction enzyme sites represented here as M, X and S. If we want to clone block A next to block B, what we need to do is that we digest the very first vector with M and S, that is 2 restriction enzyme sites, and the second plasmid is digested with M and X. Now once we ligate it, what will happen, we will have a small scar, but block A and block B will be cloned simultaneous to each other. In this case, we can have, if you want to express block B and block A with 2 different promoters, we can introduce independent promoter between block A and that promoter sequence between block A and a restriction enzyme site X, Similarly, in front of block B before the restriction enzyme X. Recombination cloning system. In this, unlike the previous three systems, we use recombination as an approach to clone our gene of interest into a vector. In infusion cloning, gene of interest is amplified using partial overlapping plasmids, and these primers have overhanging sequences which allows us to amplify a large DNA sequence. Also, we have a linearized plasmid which will have the overhang very similar to the primers which are used to amplify our gene of interest, and eventually the PCR amplified product with the primers and our vector is incubated and PCR together, which results in the cloning of our gene of interest into the vector at our desired place. Here is the schematics of how infusion-based method is used for cloning our gene of interest into a vector. As you can see here, our gene of interest, which is shown in green here, is amplified by primers which are having overhangs. And remember, these overhangs should be same in the vector where we want to insert our gene of interest. So after PCR is completed, we get a PCR product which has two overhangs, and now these overhangs, these PCR product with these overhangs are incubated with a vector which is linearized and contains very similar overhang. So these two are incubated. Now what happens inside that Eppendorf tube is that once you incubate these ones, because these primers or these overhangs are homologous or similar to each other, then there is a recombination that happens. Now in this linearized plasmid or linearized vector, instead of we will have our gene of interest cloned in it, as shown in this picture. Once the reaction is stopped, we take these reaction mixture and transform it into vector, into a bacteria or into a competent cells. Now colonies are screened based upon the antibiotic selection marker that is there in the vector. So we can screen our gene of interest and further confirm it by PCR methods. So in this fusion method, we have not used any restriction enzyme sites or restriction enzyme as such, but we've been able to clone our gene of interest in the vector. Gateway cloning. So here is a brief overview about gateway cloning. Gateway cloning is based upon site-specific recombination system of P1 phage. So P1 phage has a capability of inserting its genome or its DNA into E. coli. All it requires that E. coli, the host, should have a AdB site. So what happens in that case, the AdP site from the phage and AdP site of the E. coli can recombine and could result in insertion of phage DNA into the plasma, into the genomic DNA of E. coli. Recombination requires two integration system enzymes. One is encoded by the phage itself, which is named as integrase, and second is encoded by the host, is called HIF, integration host factor. And recombination between AdB and AdP site results in AdL and AdR sequences. This integration can also be inversed in presence of other enzymes, which is XIS, and this entire reaction can be reversed also. Gateway cloning, as I mentioned earlier, exploits a P1 phage mediated integration of DNA method in which at AdB site in the host genome, phage can introduce its DNA sequence by recombining with its AdP site. And as is shown in this picture, AdB site getting recombined with the phage DNA, resulting in two sites, AdL and AdR, and in between that, the phage DNA is being inserted. This entire reaction is called integration, where AdB and AdP sites are involved. However, this reaction can be reversed, and we can get back AdP site as well as AdB site restored, where AdL and AdR sequence can recombine and excise out the phage DNA. There are different enzymes which are involved in integration and excision processes. Gateway cloning utilizes these method or this approach for cloning our gene of interest into a plasmid. So gateway cloning is an in vitro version of AdP and AdB recombination method that we described in previous slide. And similar to that, we can also do integration into a specific locus as well as can excise it. So in order to clone a gene of interest into a specific location in the add site, there is some tweaking that has been done. So now we have named site in the vector as well as in the vector as @1 and @2, which allows the specificity in terms of its direction as well as its location. So what happens in this case, for example, an @B1 site recombines with @P1 site, and @B2 site recombine with @P2 sites. So in that way we know the location and the direction of our gene of interest. And in this method, excision as well as insertion, the reaction is similar, have similar efficiency. So we get, if we want to clone or if we want to excise our gene of interest, it can be done with the equal efficiency.
The very first reaction of gateway cloning is called BP reaction also, or in general, it means cloning of a gene of interest into an entry vector. This is the first step in the gateway cloning, where we amplify our gene of interest, which contains @B DNA sequence. Also, we have our donor vector and also the enzyme that allows the recombination process. Usually, this reaction is allowed to be carried out for 1 hour. Then the reaction mixture is transformed into the competent cells which are resistant to phase. Transformants are screened by PCR as well as using the selection markers, and then also confirmed by restriction digestion or southern blot, northern blot, or any other method which are available in the lab.
So here is an overview of BP reaction. In this schematic you can see that we have amplified our gene of interest with specific add sites. So it's @1, @2, and also in our vector, which is our entry vector, entry clone at this moment, we have similar site named 1 and 2. Now once we incubate this reaction, and what we see, there's a recombination which results in cloning of our gene of interest into the vector, but also results in a byproduct. So this byproduct is lost once we select our clone onto a selection media. And remember, only those vectors that has that restriction, that has that antibiotics marker, can form a colony on a plate, while byproduct cannot do that because it has no origin of replication. It cannot independently replicate. So we get in our entry clone our gene of interest cloned.
Now the second step that is involved in gateway cloning is LR reaction, which means that we need to subclone our gene of interest from entry clone into a destination vector, which could be expression vector. If you remember, our gene of interest was first cloned into an entry vector. But if we want to express that gene, we need to subclone it or transfer it to an expression vector. So in order to do that, we'll have an expression vector, and this expression vector will contain desired add sites. Also incubate with entry clone, which already contain the add sites, and also we add enzyme in it. We allow this reaction to go on for 1 hour, and then this reaction mixture is transformed into a competent cells, and these competent cells are phase resistant. Thereafter, we screen the transformants. We plate these cells into a selection media where we have our desired antibiotics, and these transformants are screened and clone is confirmed by PCR or restriction digestion. Here is a schematic of LR reaction which we just discussed in previous slide. As you can see, we have our entry clone, which is shown here on the top left, and our gene of interest is already located or inserted at L1 and at L2 site. Now we mix it with our destination vector. In this case it's an expression vector, which also contains at R site, at R1 and at R2. It contains also a gene in between. Now when these two are mixed, what happens due to integration or recombination, we see that our gene of interest has moved from entry vector into the expression vector, But in this reaction, we also get a byproduct in which the gene which was cloned into this destination vector or was present in between at R1 and at R2 is being transferred into a byproduct vector, and now the sites are changed from at R1, R2 to at P1 and at P2, while our expression vector will have our gene of interest with at B1 and at B2 sites. So this reaction mixture containing both of these byproduct as well as our expression clone is transformed into competent cells, and based on the antibiotics, because our destination vector contains an ampicillin in this case, we screen these transformant on an ampicillin plate. So we only screen for expression vector while the byproduct cells are not screened or are killed during our selection. Therefore, we clone our gene of interest from an entry vector into expression vector in this reaction.
And here is an example which I just talked about. So in the middle of this figure you see an entry clone, and surrounding it are different destination vector which can express in different systems. They can express in insect cell line, they can express in in bacterial system, they can express in eukaryotic system, using just at sites in these destination vector, irrespective of the restriction enzyme site availability. We can transfer this gene of interest into all such kind of destination vector. So students, let's now summarize what we have learned in this module. We have learned the different approaches of cloning a gene of interest into a cloning and as well as an expression vector. What we have also learned that cloning is not a straightforward technique. There are several factors which dictate the cloning of gene of interest into a cloning vector, and the routinely used approach in the lab is a restriction enzyme cut site based cloning approaches. Gateway cloning, on the other end, provides you a recombination based method which is independent of restriction enzyme as well as a ligation based method. Gateway cloning exploits at P1 and at B1 site, which are taken from a P-phase system to insert our gene of interest into a target site. And one of the most advantage of gateway cloning is that you can, once the gene of interest is cloned into a entry clone, that is the very first step of the gateway cloning, then it's easy to transfer our gene of interest into any destination vector, and that destination vector need not to be expressed in bacteria only. It can be used for the insect cell line, for plant cell line, as well as for yeast system or for eukaryotic system. So gateway cloning is a terrific tool to go to clone your gene of interest in a restriction enzyme free method. Thank you.